Advanced Higher Biology, Unit 3, Investigative Biology. Key Area 1, Scientific Principles and Processes. We'll mainly be concentrating on Section A of that course. So Section A covers the scientific method. It looks at the stages of the scientific cycle, and it explains why scientists discuss theories rather than looking at facts. We'll look at the null hypothesis and how even if you find that your findings don't support your hypothesis, it doesn't necessarily invalidate your theory. We'll look at conflicting data and how this is resolved between scientists, and why it's really important that we get independent confirmation of our results. Okay, so the scientific method is our way of investigating the world around us without letting our biases affect our conclusions. When a new area is being investigated, we first make observations and record what we see. Based on these observations, we construct a reasonable and testable hypothesis. We can tell if our hypothesis is testable if we can conduct the next stage of the scientific cycle, which is to design an experiment which would test our hypothesis. We carry out the experiment, recording and analysing our data before really thinking and evaluating whether our results are actually useful. If reliable and valid results can't be gained from our experimental design, we really have to think about it and redesign our experiment to better get results. We would then make our conclusions based on our investigation and consider if we need to revise our hypothesis, and again design another experiment, improve our understanding further. This is why it's a cycle. Science has never proven anything. It's just the refinement of ideas, which is the norm. Our knowledge can be best thought of as the best current explanation for what we have seen based on empirical evidence. We then look at the null hypothesis, which proposes that there is no statistically significant effect as a result of the experimental treatment. The example we use here is a zoology example, as that's what I like to talk about, but we will also touch on how this links to medicinal medicine. If there was evidence for effect unlikely due to chance, then the null hypothesis is rejected. If an investigation took place to find out if eagles had a preference for prey with red or green shells, the null hypothesis would be that there was no difference in the proportion of cells picked which were green or red. So if the researcher investigated this and found situation 1, which you can see in the table, where there's an equal proportion of red or green shells that are being picked, then they would accept their null hypothesis. That doesn't mean we've proven anything, it just means that we weren't able to disprove the null hypothesis this time. If, however, we saw situation 2, where there is a clear and significant preference for green shells in this example, we would investigate further and probably reject our null hypothesis. We'd also have to consider the validity of this experiment, as quite clearly some of the shells have 2 holes and some have 1. So it may not be a colour, it may be a size or shape or access question. If, however, the null hypothesis was looking at drug design, we might conclude that there was no significant difference between the survival of patients who were given a new cardiovascular drug versus an older established treatment. If we found that there was a better survival rate with the new drug, we'd be able to reject our null hypothesis and publish our fantastic findings. The scientific method is such that even if we don't find an effect, it's still very, very important and very, very useful to science, so long as our experiment was designed validly and with reliable design. Money is wasted all over the world when different labs carry out the same experiments just because no one is bothered to actually publish their findings. There are actually journals called the Journal of Negative Findings where you can publish these results so that they get out within the scientific community. If studies are carried out by different labs and get conflicting data or conclusions, this is okay and can be resolved through careful evaluation, or if there's still disagreement, it can lead to further experimentation. In this way, we solidify our understanding of science. Often, we find we will make discoveries that cannot be replicated by other researchers. This is often due to very small errors in our initial design or analysis of the study. This is why we treat single results that cannot be duplicated with an awful lot of suspicion.